Hi, my name is Jacqueline Hawkins, and today I'm going to be talking a little bit about Dr. Barbara McClintock. So, just to introduce her a little bit, she did attend the College of Agriculture at Cornell. Initially, it's a fun fact about her, her mother was against this idea as it would decrease her chances of getting married. But after taking her first class in genetics, she devoted her life to the study of cytogenetics, which is the study of chromosomes and their expression in the human body. Um, from Cornell, she received her bachelor's, master's, and doctoral degrees, and she mainly focused on the cytogenetics of maize. So then after she graduated, she found a spot in a research lab, and then started her research. So her focus of research was mainly on cytogenetics. So she was looking at chromosomes and expression in gene of the genes in maize. Um, she initially noticed that the kernel patterns varied greatly across generations of crop, but she thought this variation was much too large to be considered a mutation. So looking further into this allowed Dr. Stevens to conclude that genes could jump from chromosome to chromosome. So genes were able to move around within the chromosome, which she called transposing. So she kind of coined the term of transposition of genetics. Um, so with these genes jumping around within the chromosome, it allowed for the alteration of the chromosome expression, because these jumping genes, as she called them, would determine the physical output of the trait. So for example, if we had a gene for eye color that jumped around in its chromosome, we get a person that ended up with blue eyes or brown eyes or green eyes, stuff like that. Um, the issue with this discovery, though, was that it contradicted the current theory of the time. The current theory was that genes were consistent and stable, and they were passed through generations and were the same for every single generation. So her discovery that genes could change and the output of those genes or the expression of those genes could also change contradicted that current theory. So with that contradiction, that kind of led to her not publishing her work until it was proved by other scientists. So she actually waited about 10 years before she published her work, as she wanted it to be confirmed by other scientists. So once it was confirmed, she was awarded the Nobel Peace Prize. However, this was about 30 years after her initial discovery. She was awarded the Nobel Prize in Physiology and Medicine in 1983. And she was the first woman to individually receive this award. So previous women had received the award, but they had received it with a partnered male scientist. So she was the first woman to receive this individually. She also was the first recipient of the MacArthur Foundation grant, and her discovery in the field of cytogenetics paved the way for future advancements. Cytogenetics has many applicable areas. So genetic transposition is used in genetic engineering, immunology, cancer research. It's also mainly used in antibiotic-resistant bacteria. This is where other scientists were able to prove her work when they found genetic transposition in genetic or antibiotic-resistant bacteria. And there's many more examples of this, but her discovery really did pave the way for future advancements in genetic research. Thanks for listening. Here's my references, and I hope you enjoyed.